Good afternoon. So today we're going to talk about nerve function and how neurons send messages from one neuron to the other. So we're going to start today talking about the membrane potential and how that is important in the process of nerve function. So in order to send the action potential down a membrane, you have to understand that there's different charges on the inside and the outside of the membrane. We'll see this on the next slide in a moment. We look at the image that I'm going to show you. Every membrane has this polarization, and changes in this polarization are what's going to send the signals down to the end of the membrane. Once the signal reaches the end of the neuron, the synapse, then chemicals like acetylcholine, neurotransmitters, are going to be sent to be received by the next neuron in sequence. In order to get the message to the synapse, though, we have to understand the process of a resting potential and an action potential. So resting potential is important to understand because you can see from the diagram over here, there's a lot going on inside and outside the cell. Many solutes are dissolved into the solvent inside and outside of cells. So we can see a number of them here. You can see there's negative chlorines on the outside, there's positive sodiums on the outside, positive potassiums on the outside as well, but more positive ions on the inside as well. You can see positive sodiums, positive potassiums, and then negative proteins. When we kind of tally all of these positives and negatives up, what we find is the membrane is more negative on the inside. It actually has a voltage of negative 70 millivolts. One of the quick ways to remember where the ions are is by remembering what we call the salty banana. Okay, so salt we always think of as sodium. That means there's more sodium on the outside, right? Salt on the banana. Potassium, which we know bananas have a lot of potassium in them, that means there's more potassium on the inside. So whenever we talk about that negative 70 millivolts, the resting potential, that is what we're going to refer to, remind you, with the salty banana. Now, how does this change? You can see in the diagram that the ions are kind of always moving. The potassium is always trying to leave the cell, sodium's always trying to get in. And then in the middle, you've got this sodium-potassium pump that's kind of forcing them to go in the other direction. All of this is going to impact how those messages are sent and how those action potentials are sent. So the resting potential, again, is negative 70 millivolts, and that tends to stay that way. If that changes, then it can go into what's called an action potential. This is where the ions quickly change locations and cause a change in the polarization of the membrane. So the first thing you have to understand is a stimulus will happen. If that stimulus goes above negative 55 millivolts, that is above what we call the threshold, that will start the process of an action potential happening. And what's actually going on is that will cause the sodium ions to rush into the cell, which we call depolarization. The sodium ions will rush into the cell until we reach between positive 30 millivolts and positive 40 millivolts. Once we hit that peak of between 30 and 40 millivolts, then those sodium channels will close and the potassium channels will open. And that will cause what we call repolarization. So we're getting back down to that resting potential that we had before. So the sodium is kind of now on the inside of the cell and the potassium is leaving the cell. That will continue happening until we get to, again, below our resting potential, which is negative 75 to negative 80 millivolts. The problem is our ions are not in the right place now, right? Our sodium is now on the inside and our potassium is now on the outside. So what we need to have happen is we need to get back to that resting state, the resting potential of negative 70, and the ions in the right spot. So the sodium potassium pumps come along and they push the ions back to where they need to go. Anytime from threshold until we get back to that negative 70 millivolts, we call that the refractory period. Okay. Just a couple of things to sum up with you. As you guys know, we talk a lot about transport in the cell. So active and passive transport are both involved in the membrane potential. Ions are going to move through their ion channels to go where they want to go according to the gradient. So sodium wants to go inside, potassium wants to go outside. But again, that will be negated by the sodium-potassium pump. So we can see that if the sodium channels open up, they want to go inside. That'll cause depolarization. When the potassium channels open up, they want to go outside. That will cause repolarization. And then the sodium potassium pump kind of gets everything back to where it needs to be. Next time, we'll talk about how this proceeds down the entire length of the membrane.